The title of the topic, Study of Eco-Friendly Biocontrol of Penicillium Fruit Rot of Citrus reticulatus by yeast. Citrus reticulatus is the orange fruit. Okay? And you are seeing the orange plant. Actually, it is a green technology, post-harvest green technology. And the orange are there. Orange is very important fruits. It is delicious and it is full of vitamin C. But its dangerous disease is Penicillium vegetatum, fruit rot of this fruit. And it is a post-harvest disease. This disease causes from 40% to 100% damage of orange fruit. This disease is generally controlled by application of fungicides which are causing health hazard and environmental pollution. Moreover, some microbes becomes fungicide resistance and it is very dangerous for environment. Modern trade is application of yeast as biocontrol agents. Many post-harvest diseases of many fruits and vegetables. It is the modern yeast. What are the yeast? The yeast are unicellular fungi and they are non-pathogenic and saprophytic. Therefore, the main objectives of this work are to control post-harvest disease of orange caused by Penicillium vegetatum by three yeasts. Three yeasts are Candida hamata, Deberomyces hansensi, Rhodotorula miscellaneosa. No use of chemicals, no use of pesticide. It is a green technology. In the left side, we are seeing that fruit is just attacking by the fungus pathogen and form a white cotton. In the right side, the disease become mature and whole orange fruit is damaged. And we isolated the pathogen. We isolated the pathogen from the diseased fruit, and the left side down is in the culture, pure culture of the pathogen. And in the right side, it is the Penicillium vegetatum under microscope. When you view under microscope, this is the structure. And this is the two yeast. One is Candida hamata under microscope. Extreme left, it is the culture in slant culture. Middle, it is the under light microscope. And right, it is the under scanning electron microscope. And it is another yeast which is Deberomyces hansensi in slant culture, and right is under light microscope. And it is another yeast which is Rhodotorula miscellaneosa, and under light microscope it is extreme light. For eco-friendly control of this pathogen, we used three yeast, Candida, Deberomyces, Rhodotorula. Two methods are taken here, laboratory method and large scale method by surface wound method. Actually, it is a green post-harvest technology. Laboratory method and large scale method. First, orange are washed with tap water, then air dry. A triangle is made at equator zone with sterile needle.
the pathogen and yeast are inoculated in the same injury of the orange, and large scale fruits are deep in suspension of both. Incubated the fruits in trays covered with polythene at 26 degrees centigrade for 7 days. What result? In the left side, we treated Candida fumata, the yeast, deep, with Penicillium digitata, which caused the fruit. But in left side tray, there is no disease. Remember, in the left side tray, there is no disease. But in the right side tray, the fruits are attacked by the Penicillium digitatum and form greeny coating. So, it is interesting to say that using the yeast Candida fumata, we can totally control the pathogen without any pesticide and fungicides. These are also result from the Rhodotorula mucilaginosa. In the left side, some of the fruits have been attacked, but in the right side, maximum fruits are attacked. So, it was found that if we use 10 to the power 8 cells per ml of Candida, the cells contain concentration, we can control the disease without any pesticide and fungicide. So it is the green technology. What are the disease? 12.45%, 20.11%, 25.23% of disease exists. So near about 70%, this can be controlled without using any pesticide and fungicide. In vitro interaction, if we found how they control the disease, the pathogen, interaction, in the laboratory, we found that in the right side, the larger filament is the Penicillium digitata. Each cell overpowered them. Each cell overpowered them. In the left side, in the petri place, the Candida fumata secretes some antibiotics or other things. As a result, the mycelium become damaged and form a clear zone. It is the scanning electron microscope. Whenever we introduce in the same injury the Penicillium digitatum and Candida fumata, after 24 hours we found that each cell colonizes very quickly and dominate over the Penicillium digitatum. It is the after 48 hours. After 48 hours, when we scan under electron microscope, that Candida fumata moreover dominate over the injury. And we found that some volatile substances are also coming from the yeast culture, and these are the some methods to identify. And this is the percent of disease incident I have already told. It is also percent. And conclusion. In conclusion, the yeast cell Candida fumata 10 to the power 8 cells per ml can be applied to control the orange rot caused by Penicillium digitatum. And we are facing fund grants for more improvement of these innovative green post-harvest technology. More funds are required. Any kind of it or any interested candidate or company may come forward for the benefit of common people in form of donation or collaborative work. Thank you.